let's start. Sorry for running late. Another shoot went over time. Okay, so the last lecture for our protein targeting. Excellent. Okay. As always, learning outcomes. So this is what I want you to get from these lectures, okay? Well, actually understand what the term sorry. Hang on. I've got to switch this off. I've got to switch the Wi-Fi off. Otherwise, you'll be getting notifications all throughout the lecture. Okay, so I want you to be able to actually define or understand what the term mitochondrial biogenesis means. Understand why it's an important area of research. Get it? Import? Okay. So really, this whole lecture is going to be about the methods that we use to study protein import pathways and components, basically what I do in the lab. We're going to talk a little about how knockout lines can be used to study protein uptake. So this is using generally reverse genetics. Yeah, reverse genetics when you make mutants of a particular gene to understand its function. We're going to describe methods to assess, or does that say access? Then we're going to have factors that can regulate mitochondrial protein import. That's the last sort of thing. Okay. Right, so why do we study protein import? Because it's known to regulate mitochondrial biogenesis. So what is mitochondrial biogenesis? So mitochondrial biogenesis is just the change in mitochondrial mass and number. Yeah, so you have a certain number of mitochondria in the cell. That mitochondria can increase in mass, so it can uptake more proteins. Yeah? So that is why protein import is important, because it obviously needs to import all of its proteins. But it also refers to mitochondrial number as well, so you have division, a fusion of mitochondria. But we all know that mitochondria are really important for energy production. Yeah? You've been told this from day one. So that's why mitochondria is really important in so many diseases. So even though I work on plants, mitochondrial diseases are really important, right? And a lot of defects or mutations in mitochondrial protein import components and apparatus result in serious mitochondrial diseases. So a lot of medical researchers are working on very similar pathways and processes, but all in human cell lines and things like that. Okay, so so not just mitochondrial encoded genes, so even mutations in mitochondrial proteins that are nuclear encoded. Okay, it's important. Okay, so why do we want to study these, sort of the mechanisms of imports? Well, one method to cure a lot of diseases is to, replace that non-functional gene with a functional one, right? So if you can deliver DNA or RNA or mRNA to the mitochondria, you can then sort of complement that mutation, yeah, override that mutation. Or if you have a defective protein in the mitochondria, if you can target a non-defective one, then you can obviously complement that function as well. So that's why studying these mechanisms of targeting and trafficking throughout the cell, can be important for sort of possibly then, finding cures for a lot of these diseases. So gene therapy is quite popular for a number of mitochondrial genetic mutations and diseases. We also want to understand how we can deliver drugs. And obviously I also mean protein and drugs. I mean peptides, protein, DNA and RNA, yeah? Not just chemical drugs. Okay, so the definition of mitochondrial biogenesis, well, in plants and actually any system, so apologies for having implants, but it's for everything. It's the increase in both mitochondrial mass and number. So traditionally we only talked about protein import when we talked about mitochondrial biogenesis. But now, of course, we know that mitochondrial biogenesis also is regulated by gene expression. Yeah. It's also by protein import, obviously, but also a new sort of more area of research is looking at protein degradation. So how are the proteins that are already imported into the mitochondria, they're already in there and functioning, what's their turnover? Yeah, proteins have a lifespan. a lifetime, and different proteins are getting turned over at different rates. So it's really interesting when you look at degradation of proteins, yeah? Like even within one complex, for example, let's say complex 1 on the mitochondrial respiratory chain, really important complex, it's the site of electron transfer. Some of the components are turned over much faster than the others. So how is that regulated? It's kind of amazing, yeah? One component can be there for, let's say, well, I'm not saying years, but let's say it could be there for days, yeah? If we look at, protein turnover and lifespan. Some proteins can be there for days. Other ones in the same complex get rapidly turned over because usually they get degraded or they're sort of exposed to ROS and they get damaged a lot faster. So it's an interesting area of research. How are these proteins degraded? Why are they not all degraded the same way? What proteases do this? Yeah. Okay. Right, so the degradation of mitochondrial protein, and we have to look at the degradation, of course, not just normal life or development, but also when at times of stress. Yeah, it's going to happen more when plants are stressed or human cells are stressed. You have more ROS, or you need more energy production, so you need to turn over these proteins. Yeah, they get used up faster. Okay, so this is basically just the same slide, just summarized, that mitochondrial biogenesis is defined as changes to mitochondrial mass and number, and all these factors can regulate mitochondrial biogenesis. Okay. And what's important is because we're talking about protein import in this lecture, it's really important to know that protein import is a regulated process. We've noted it's regulated over development. There are different tissues that import proteins differently, right? And it is stress responsive. So when a cell is stressed, it completely regulates, it changes the rates of import, of protein import. So we're finding out that there's a lot more sort of intricate mechanisms of regulation. So it's not just as simple as identifying what are the receptors, what are the transporters. It's more going into now, well, how are they regulated? Yeah? What changes the abundance? Okay. So this is sort of a dot point 1 to 6, sort of how we study mitochondrial biogenesis. So the easy part is to, of course, identify and characterize a component. Yeah? You can identify a receptor. You can identify a receptor or a transporter based on homology. Luckily for me, there are a lot of people that work in yeast, a lot of really good labs that work on protein import mechanisms in yeast, and every couple of years they identify a new import component. So they do all the hard work. I don't have to do, because I look at the protein that they've just identified, and I look in the plant genome, and I say, look, plants make one too. So that's how I can identify new import components. It's not that easy. That's lazy, no. It's not lazy science. So identifying is always on homology, yeah? We know that proteins are conserved, functions are conserved, and plants, animals, fungi, whatever, protein import pathways are really strongly conserved because they're an ancient bacterial pathway. Yeah, they're really strongly conserved and maintained. So I can identify import components purely based on homology. But I want to know, well, okay, so that's the easy part. You can then knock it out or overexpress it and look at all the effects of doing that. But sort of the hard question is, well, how is this whole process regulated? We know that the abundance of particular transporters directly correlates to the rate of import. So I've shown, and other people have shown, that if I increase the abundance of just TIM17, I can increase the rate of import twofold. Yeah? So it's directly correlated. But we also know that there are a lot of cytosolic factors involved in protein targeting. Yeah? We have a lot of chaperones that need to deliver that protein to the mitochondria. We really don't know anything about which cytosolic factors, how are they regulated, how are they specific for their binding, do they have specificity? Right? But we know that they need to be there to maintain these proteins in an import competent state. We're also finding out that, well, there's a lot of interactions between the import translocases, yeah? The outer membrane receptors or the channels actually interact with the inner membrane channels. And that makes sense, right? You want to be efficient. You want that protein to get targeted across the two membranes. You don't want it floating around anywhere in the intermembrane space. You want it to be efficient and get translocated or trafficked to where it needs to go fast. And we're finding more and more examples where there's interactions between the TOMs, the TIMs, the SAMs, the TOMs, yeah, even between, it's the MINOS and the ERMES I may have covered, between the ER, right? So you can get shuttling of calcium. But there's more and more examples where we're actually finding out interactions between the respiratory chain and protein import apparatus. So the respiratory chain is, let's say that's the main function of the mitochondria. It's not the main, but it's a really important function. And it needs a lot of proteins. All the complexes 1, 2, 3, 4, 5, they all are made up of multiple subunits. So we need a lot of protein import for those complexes to get assembled. So we're finding out more and more examples where these TIM17s are interacting with respiratory chain complexes. Yeah? And if you remember how I talked about MPP in plants, the mitochondrial processing peptidase, that actually is complex 3. So it doesn't have an interaction, a physical interaction, but here we have a protein that has two functions, import and respiration. So there's more and more examples of this happening. Now, the last one is something we're trying to really delve into is, well, what are the signals? We know that the mitochondria, or we know organelles signal, yeah? It's called retrograde signaling, where the mitochondria, the chloroplast, or the peroxisome sends off signals to the nucleus, so you can modify expression, you can initiate the translation or gene expression of a particular factor that's needed, yeah? It's always communicating. So not just between the mitochondria and the nucleus, the mitochondria and the chloroplast communicate as well. So it's a pretty amazing network of signaling going on in the cell. Okay, so here's the so somebody else has published a really nice review paper on approaches that you can use to identify and characterize mitochondrial protein import machinery. And I don't use all of them, but a lot of these we actually do use, right? So first of all, I always look at homology, yeah? So it's an easy way to identify new import components. Look at homology of something that's already been identified or has common domains, yeah? So, for example, if you look at the TIM17s and the TIM23s and TIM22, they have a conserved motif, yeah? And so this has led to the identification of new import components because that motif, that GAA whatever, is conserved across different species. Another good method that a lot of people use, and this is a non-biased method, is doing pull-down or interaction studies. You tag your transporter or receptor, and you pull it down, and then you identify, well, what does it interact with? What other proteins do you pull down with it? So that has led to a lot of identification of new receptors, new components, ones that you wouldn't come across by looking at homology, yeah? Because it's a non-biased method. You don't know what you're looking for. So you can come across a lot of new and exciting receptors and components this way. Of course, the most common method has been doing knockout lines. So, we have yeast. Have you heard of the Saccharomyces deletion project? So yeast is our model species, yeah, eukaryotic species, but you can obtain a knockout for every single gene. So people do that. They look at the knockouts and they screen these mutant lines and they try and identify yeast that are respiratory deficient, which means that they don't grow so well on non-fermentive media. So we know it's a mitochondrial defect because it's related to their respiration. But they kind of screen thousands of mutants to identify mitochondrial defects. And a lot of import components have been identified this way. Okay, so we can also use mass spectrometry to identify substrates. So this is also with tagging methods, yes? So if you tag it, pull it down, then you can identify, well, what does the receptor bind to? It's probably not that great because we expect that a substrate and a receptor interaction, so a precursor protein binding to TOM20, We expect that interaction to be quite transient because it's basically only it's only related to that peptide binding, so it's transient interactions. Okay, so once we've sort of identified an import component, we're then going to confirm its location. And I'm an import lab. This is what I mainly do, protein import assays. So I will import it to see, does it import into mitochondria? That's one way of confirming its location. Another way is obviously to do immunodetection. Do we have an antibody for it? Then I can detect to see if it's mitochondrial. I can also look, where is it in the complexome? So, proteins usually interact in larger complexes, especially import components. So does it interact with the TIM1723 translocase, or does it interact with the TOM complex on the outer membrane? Okay, and then once we've shown it's mitochondrial, we can see where it interacts or what is it interacting with, is it in a complex, we then want to characterize its function. And that can be hard because you've got to look for phenotypes, yeah? So you know what a phenotype is? An observable defect. And this way generally we obtain knockout lines for our gene of interest for encoding our protein. Or if we can't obtain knockout lines, we overexpress it. Or now, I guess, if we don't have knockout lines, we CRISPR it, or RNAi, yeah, so we can get knockdowns. Okay. So generally with knockout lines, you generally can just purchase knockout lines for whatever gene you want in yeast and plants. Okay, and then we study its phenotype, yeah, this is the consequences of defect. Does it grow normally? Does it look happy? Yeah, does it import proteins? Okay. So how do we identify a putative homologue? So it's always a putative homologue, yeah? If you do anything, like if you do BLAST, you would have done a BLAST somewhere in your undergraduate, yeah? You're just looking at homology of DNA or homology of proteins. So you just BLAST to identify a homologue. And it's always putative. Okay, so we BLAST to identify something, and there's pretty much all genomes available for nearly every species now, yeah? I think we have annotated genomes for pretty much everything. We can look at its phylogenetic analysis, yeah? So when did this protein evolve? Is it conserved? So generally, if it's present in every single species, we know it's important. That's always a good sign. Yeah? If that protein is present early, and so of course in plants, which is a good model to use, you can look at the evolutionary age of plants, yeah? You can have green algae, so the earliest form of plant, then you have land plants, angiosperms, so you can actually trace the presence of your particular protein throughout evolution, yeah? And you can go back to millions of years, whatever. And if it's arised early, you can sort of be confident that it's an important fundamental import component, like a transporter. Yeah? It's really fundamental for its function. Or it's essential, yes, because it's always maintained. There's no gene loss across evolution. So a lot of people just do bioinformatics based on phylogenies and, just computer programs, and it can still tell you a lot. So did they arise, did they arise early? You can also look for targeting signals. So I've mentioned to you that there are a lot of online databases where you can predict if your favorite protein has a mitochondrial targeting signal. So these are based mainly on the fact that there are those certain characteristics I talked about targeting peptides. Yeah, we learned about the -2 arginine rule. We learned about alpha helix. So all those characteristics of targeting peptides you can predict. Okay, so we want to see if these proteins are likely to have a targeting signal, but of course remember that only 70% of mitochondrial proteins do contain a targeting signal. So it's not definitive. And you always have to be dubious about predictions like this. Yeah, because how was this prediction model built? Is it going to cover my species? Is it going to cover inner membrane proteins? Okay, and then you can always look at the protein length. Does it have conserved protein domains? Are there any unusual features? So this is kind of really how you study any protein, to be honest, in any kind of research area. You'll be doing pretty much the same things. Okay. And always go into the literature. There have been many occasions where I started working on a protein thinking, oh my God, I've identified this is fantastic, oh my God, this is going to be great. And then, a year down the line, oh wow, it's already been published. So you have to look, just look, really. You'd be surprised, because it may not have been annotated as an import component. Or somebody just identified it in completely different, context or biological process, yeah? But they've identified it's already been published or whatever. So you have to be really careful. All right. So for example, somebody might have identified an EMS mutagenesis screen. So it's a completely non-biased way of looking at identifying protein function. Okay, but not actually looking for protein import. Okay, so once you've decided you're going to characterize a protein import component, you're going to spend the next 3 years of your PhD doing this. You're going to clone the gene. And that's the easy part, because you can just do a standard PCR from, total cDNA using gene-specific primers. If you're really lazy, you just order clone. Or if it's really hard to PCR. Okay, some genes are just really troublesome for whatever reason. But usually it should be easy to clone your gene. Then you want to determine its localization, right? Where is it targeted? So the first thing I would do is actually just put a GFP on the end of it and then transform cells to see where it's going. Then, once I've shown that it's mitochondrial, I might actually do protein import assays, and I'll talk about those later. Okay, so here's an example of a phylogenetic tree of import components, right? So I told you about the TIM17, TIM23s, TIM17, TIM23, TIM22. They're all related, and they're all actually defined as pre-protein and amino acid transporters. So they're kind of a big family of proteins, and they don't just import proteins, they import single amino acids as well, and maybe even metabolites. So it's good, because I've been working on PRATs for like 15 years, 20 years. So you have TIM17s, TIM23s, but what's interesting is we also have PRATs that are in the chloroplast, right? How does that work? Because I told you that chloroplasts, I showed you how chloroplasts import their proteins, and they don't have any TIM17s or TIM23s, right? They have different ticks and tocs. They're completely unrelated transporters. So how are these proteins that are really closely related to TIM17, TIM23, and TIM22, how are these in the chloroplast? Well, these proteins actually are in the outer envelope, and they transport metabolites. All right, so that's quite cool. Now, there are some down here, these ones, we published on these last year, the year before. They're actually in both organelles. So they're in the mitochondria and they're in the chloroplast, to add to the confusion. And they don't import proteins, they don't import metabolites, they actually import tRNA. So a completely different function, but still related, right? So it's kind of amazing how this family of proteins, these genes, have evolved. They've acquired new functions. Well, they've modified functions based on their localization. So there's still a few more of these to find out what they do. Oh, okay, well, here's actually a summary. I forgot about this slide. So here's a whole gene family of import components, right? TIM1723 we have here on the inner membrane. And even within this gene family, we've shown that one of these, TIM17, actually is germination specific. So it only is present in a dry seed. And it only is expressed during germination. And it's only expressed to increase mitochondrial import at that small particular stage. So it's kind of cool how we can identify sort of different functions. And here's the tRNA import receptor. Here we have TIM23, also an important transporter of the inner membrane. Don't panic, you're not going to be assessed on this, okay? Sorry, this is just to give you an example how you have a lot of divergence in function, and especially in plants or higher eukaryotes, yeah? There's multiple genes that encode for a particular protein, and even these multiple genes, and they encode for related proteins, they can have different functions, and quite diverse functions. All right. Okay, so how do we study proteins localization? Everybody does GFP tagged. Yeah, it's The most common procedure, GFP tag your protein, see where it goes under a microscope. Some people will go ahead and do protein import uptake assays, not many because you have to use radiation and people don't like that. If you're really dedicated, you're going to make an antibody and then you're going to do a Western blot. Yeah? So you guys, have you done any Western blots in any of your labs? So it's kind of a straightforward process, yeah? Oh, but all these raised eyebrows and not successful Western blots. So if it works wonderfully, it's wonderful if you have a beautiful antibody. Yeah? If you don't have a good antibody, it's horrendous. But sometimes it's worth spending that time to make an antibody. So you would have to clone the gene. You'd have to express the protein in bacteria. You'd have to isolate and purify your protein using columns or nickel chromatography. Then you have to go and inject a poor bunny rabbit with your protein. And then collect blood over 6 weeks. And then that sera is what you use for your Western blot. It's antibody. Yeah? So you don't even have to purify this from the serum. You just use that serum straight onto a Western blot. So that's how most antibodies are made for polyclonal antibodies. Okay. Then if you've definitely confirmed that your protein is where it should be, you would go ahead and make knockout lines or make overexpressors. Okay? Right, so GFP tagged. So you probably would have come across GFP tagging and localization in previous lectures, but what I always test is you can test both termini, N and C. So do you remember how I mentioned in peroxisomal targeting, peroxisomal import, you can have a C-terminal PTS1 signal, yeah? So if you have a C-terminal GFP, that's going to mask that targeting signal. So that's why it's a good idea to try both orientations. But you don't really do that if you suspect it's peroxisomal or it has an N-terminal. I mean, generally, if I'm only interested in mitochondrial proteins, they generally just have N-terminal signals, N-terminal targeting peptides. So then I would just do a C-terminal. Okay. And you can either just deliver that DNA, the gene, yeah, your clone with GFP, you can just either shoot it into the tissue using a bialistic gene gun. So this is just, the DNA just gets coated on gold particles and you just bombard that cell. And it gets through the cell wall and that gene is transcribed and translated and you make the protein. You can also make protoplasts. Have you heard of protoplasts? So that's just when we actually just take away that cell wall. Yeah, what makes plant cells special is they have a cell wall, yeah? Plant cell wall. And you can remove that cell wall and then you can deliver DNA or anything into that protoplast quite easily using just PEG. What's that? Polyethylene glycol. So you can make protoplasts. So here's an example of protoplast. Or if you really have a lot of time on your hands and you really want to play around, you can actually make a permanent transgenic line. Yeah? So you've seen mouse models, yeah, of glowing GFP mice. Here's just a glowing GFP Arabidopsis. Okay, and then you can actually just microscope individual cells within that. I mean, this can take a year or 6 months of your life, so do you really want to do that? But the good reason about this, one of the benefits of this is you can actually clone that GFP, your protein, and GFP, but you can clone it under its native promoter. So that's the promoter that it naturally has. All these methods we, artificially express. So all these methods aren't at normal cell abundance, yeah? Using this method, not only can you look at the abundance of a protein, but you can also see where it's expressed. You might have tissue-specific expression of your particular protein, and you can do that by using transgenic lines. Okay, so the next really important method is organelle isolations and import assays. So if we want to, in a test tube, import a protein into a particular organelle, mitochondria, chloroplasts, peroxisomes, we can do all three organelles, yeah? Even nuclear imports. We have to isolate the organelle first, and this is pretty straightforward because it's just kind of biochemistry. You just homogenize and you centrifuge. So it's just a number of stages of homogenization and centrifugation, and then you have to use a gradient. Yeah? So a gradient will separate organelles based on density And there are a number of different gradients you can use. So there's a continuous, which obviously means a continuous gradient. Discontinuous, so you have strict sort of concentrations of various things like sucrose or, or PVP. So various compounds that will affect the mobility. And discontinuous, so that means you sort of spin it down first. So in each of these gradients, your organelle is going to resolve at a particular stage. Yeah, so for example, here we have mitochondria, here we have peroxisomes. In this case, it's not very good, but it's here we have mitochondria, here we have chloroplasts. Yeah, so you can isolate your organelles, really quick methods, good old biochemistry, and we can use a range of different tissues, but we need a lot of tissue, generally. Yeah, so we can use seedlings grown in water cultures, we can use just grown on agar plates, we can use little plants grown on soil, or obviously cell culture, because we can get a lot of organelles from cell culture. Once we isolate the organelle, we're going to import the protein. So the first thing you have to do, obviously, is you want to clone that gene that you want to test. We're going to clone it, and we want to translate it. So you have a lot of kits available to you where you can translate that protein in a test tube. And basically, we use a cell-free system, a TNT per mega, and basically it translates the protein in rabbit reticulysate. But what it does is that at every methionine, you have a radiolabeled at 35S methionine. So every methionine within that protein is hot, is radiolabeled. And that lets you detect that protein on a gel or on a plate. Okay, because you need to visualize these proteins. You can translate proteins, but they're usually at low abundance. Right? So you want to visualize your protein. So we use radiolabeled proteins. It's not very radioactive, so it's not scaringly radioactive, so you don't have to use, shields or anything. It usually doesn't penetrate through plastic, but it's still radioactive. Okay, and then basically, you're just going to incubate your protein, your radiolabeled protein, with your mitochondria. Straightforward. You just import it in a buffer with all the goodies, like ATP, GTP, right, sucrose, methionine, all these goodies that the mitochondria like for import. You just incubate them. And what happens is when you incubate your protein with mitochondria is you'll actually get import, and then you get cleavage of that targeting signal, that mitochondrial targeting signal. When MPP cleaves it, what happens is that you see a shift in size, yeah? It's smaller. So that's how you can visualize when it's actually been imported and processed. So this is what we see in lane 2. Our lane 1 is just precursor protein on its own. It's just the radiolabeled protein without any mitochondria added. Lane 2, we have our protein plus our mitochondria. And so here we have unimported protein. So there's a significant amount of protein that didn't get imported into our mitochondria. Here we have cleavage of that mitochondrial targeting signal, and you see a smaller size product. But what we also do is we also have another lane where we actually add proteinase K. So this is protease. We add it to the reaction. It digests all the proteins that haven't been imported. Yeah? So it digests here, but the protein that is imported and targeted and inside the mitochondria is protected. So then you have that corresponding band, okay? So this is a really easy import assay to show that your protein is inside the mitochondria, has been processed, and is PK protected. So any of you guys remember how I said we need what's one of the most definitive requirements for protein import? It's a membrane potential, yeah? Without a membrane potential, nothing gets across the inner membrane. So here we add valinomycin. Okay, so this is just a chemical ionophore. It actually dissipates the mitochondrial membrane potential. So it's your negative control. Okay, so that's just a regular protein import assay into isolated mitochondria. And this is the same assay people in mammalian cell lines use, yeast people, plant people. We use the same assay, pretty much the same things. But it can get a little bit complicated because remember, not all proteins have a mitochondrial targeting signal. So how do you confirm a protein is imported into the mitochondria when it doesn't have a targeting signal. It's not going to get cleaved. Okay. So here's a really good example. So if you remember, a lot of the inner membrane proteins, the carrier pathway, yeah, they don't have targeting signals, but they have internal signals, okay? So a lot of proteins have internal signals. So to show that it actually has been imported into the inner membrane, what you need to do is after import, you need to rupture the outer membrane. So you osmotically shock the mitochondria. After your import reaction, you shock the mitochondria, the outer membrane ruptures. If you remember, the outer membrane isn't very, it's quite impermeable, yeah? Sorry, it is permeable. So the outer membrane ruptures, that leaves just the inner membrane. And these are called mitoplasts. Okay? So if you rupture the outer membrane, you can then PK treat the mitoplasts. So then you're digesting any proteins that's in the intermembrane space. And anything that's inside, in the inner membrane, is going to be protected. So here we have TIM23. This is the portion of the protein that is protected in the inner membrane, and PK can't chop it up. Okay, so that tells me that TIM23 has been imported. It's in the inner membrane, and there's a portion of it that's PK protected. Okay. All right, so but the definitive method of confirming protein localization will be to do a Western blot, yeah? So antibodies is really the most the main, because you can have mistargeting. There are many examples where I can translate a protein in the radio rabbit reticulocyte. I can incubate it with isolated chloroplasts, and it will go in, but it's not a chloroplastic protein. Yeah, so we have a lot of mistargeting, and a lot of it is forced. Yeah, these are in vitro experiments. So if you just, what's the term? I shouldn't say this, but if you throw enough shit at the wall, it will stick, right? So if you're forcing a reaction, you've got enough protein and organelle, you might get import, but it's not real. So you have to be really careful. Okay, so there are many examples where some proteins are just loose. They'll go anywhere. Right, so antibody, the most definitive control. But of course, we've done Western blots before, right? You can have a lot of non-specific bands. So how do you show that that band that you can pick up in your isolated mitochondria is your protein? Well, you have to have knockouts, unfortunately. Yeah? And then you have to do the knockouts alongside it. And only then can you be 100% sure that your protein is inside that organelle. We can then do a lot of cool things if we have an antibody, because we can then shave the outer membrane with different amounts of proteases, PK or trypsin. We can shave that outer membrane, and then we can use our antibody. And it's kind of cool. So if you look at this protein, this is also one of those PRATs that I talked about, and it actually sits on the outer membrane. So if we add different amounts of PK, we actually see that this protein gets clipped. Yeah? Little bits of it get clipped off as we go deeper into the outer membrane. And you can do inner membrane and then matrix controls. Yeah? So it's kind of neat. So you can sort of chase the localization of a protein. All right, we've only got 5 minutes. So I'm going to go through a research paper. I will go through it, might be a little bit fast, okay, because it's got big figures. But I just want to show you how, in all these experiments, how we used it to characterize TIM17-1. So it's one particular TIM17, and how we found that it has a role in germination. Okay, so first thing was to clone it. Okay, I kind of knew it was TIM17 because it's so homologous to all the other TIM17. So there was no surprises there. Okay, we radio-labeled it, and we did an import. Now, you might go, oh my God, that doesn't look like the previous panel. Because in reality, a lot of proteins have multiple methionines, okay? And when you translate a protein, it doesn't know which one's the first methionine. So it can translate a protein from the first methionine, the second methionine, the third methionine, yeah? And all these different bands represent TIM17-1 or truncated forms of TIM17-1. And of course, the full length, the biggest one is our full length protein. So here we have done an import of TIM17-1 into mitochondria, and we made mitoplasts to remove the outer membrane. And then we can see that we have a small inner membrane protected fragment. So that tells me that this protein is in the inner membrane. I then did GFP and co-localized it to with a mitochondrial marker. Oh, that's 2016. It wasn't that many years ago. But anyway, oh my God, I just realized there's a few green dots that don't co-localize. I might have to repeat that. Isn't that funny? Look at that. I mean, it should be 100% localization, right? Sometimes you get a GFP where it doesn't target at the same rate as the RFP control does. So the RFP is just a known mitochondrial control, and they are different proteins, and they have different tags, so sometimes they might not target at the same rate. But that's weird. Anyway, I also did an import into chloroplasts and show that it didn't import. Whoopee, didn't import. It wasn't expected to import. Okay, then I looked at expression, right? Because we wanted to characterize this protein. I'm like, oh God, there's 3 of them, what do they do? So I thought, well, let's have a look at the expression. And this one was really weird, because if you look at all the TIM17 ones, so you have TIM17 1, 2, 3. So this is a heat map looking at just publicly available expression analysis data. We found, well, this TIM17 one's hardly expressed. So blue means no expression, right? It's not expressed. Why is it even encoded? But if you look closely, it was mostly highly expressed in the seed. So we thought, oh, maybe it has a role in germination, yeah? The seed is important. When you have a dry seed, you have really small mitochondria. There's limited number, and they're really small. So it's a really nice stage to look at biogenesis, because in a really short space of time, you need to make a lot of energy, yeah? And the chloroplasts aren't made yet. It's not green. So the mitochondria is really responsible for driving germination and seedling development. So it's a really important developmental stage. And it seems that this TIM17-1 is only expressed in germination. So we did some more QRT-PCR and found, hang on, it actually goes down throughout germination. So it has to be present in high amounts early. And then, of course, I wanted to confirm that it's present in mitochondria, and I did a Western blot. And I swear to God, this took so long to get, because this is such a low abundant protein. I don't think I'm ever going to be able to repeat this again. Not that I've had to. But it took me forever to detect this protein in mitochondria. And the only way I could detect it was when I UV stressed the seedlings. Oh my God, like, it's ridiculous. But looking at online in the bioinformatic data, I could see that UV treatment of plants induced my protein 30-fold. So well, hang on, if I treat my plants, maybe I'll be able to detect my protein. And I did in Colombia, and not in the knockouts. So this really confirmed that, this is the band that we're looking at, that little thing down there. So this really confirmed that I had knockouts and my protein was expressed. Right. So then, like I said, we characterize knockouts. So these are publicly available lines that you can buy. I didn't have to generate these, but we had to characterize its development, right? So it's kind of tedious process where you measure root growth and developmental stages. It's not really that fascinating, except if you just look at germination. There was a significant, that's significant, that little dot, right? That little star. It means there's a significant difference at that stage. And that's, of course, germination. So we kind of broken it down to hours, right? I had to look under a microscope and count the number of germinating seeds over so many hours, and we found that our knockout lines germinated faster, okay? So when I talk about phenotypes, it can be really hard to identify a phenotype. You think, oh, you knock out a protein. It's an important protein. It's going to be sick, but it's not. Because you know what? Plants are really resilient, hey? They have other things that can compensate for deleting that protein. So that's the problem. Anyway, I'm not going to go through more details of this paper because I don't think you need to. If you want, you can go through it, look at it yourself, but it's kind of detailed and maybe like already falling asleep. But what I really wanted to, show you out of that paper is that we have to use different methods to functionally characterize protein import components, yeah? Now, if any of you guys noticed, saying, oh, hang on, so I've done all this. She did all this, right? She characterized, shown its mitochondrial, characterized the mutants, showed that they have a germination phenotype. What did I not do? What's the most important thing I didn't do? You know what? I did not actually show that these mutants didn't import proteins, yeah? So that's a big gap, and luckily no reviewer noticed that because I never actually showed that affecting the abundance of an import receptor affects the rate of import, okay? So what I should have done is I should have actually looked at the import rates of my wild type and my mutants and compared the two, yeah? The rate of import should have been different. Right, okay. Oh, regulation, sorry. Two more slides, okay. So like I said, these factors are regulated, and it's usually abundance, but there can be other various things that regulate protein import, okay? You have external signals, development, stress. It all affects the rate of protein import into a cell, okay? And I think this one's probably a little bit better, this slide, okay? So here we have sort of the main things. So the transcription rate in the nucleus, how fast a mitochondrial protein is synthesized, is translated and transcribed and translated and imported, yeah? That affects mitochondrial biogenesis. We can also get targeting of mRNA to the surface of the mitochondria. Okay, so that's quite a new sort of research where if you want really fast imports, you don't have that protein floating around in the cytosol. You actually get targeted mRNA delivery. Okay, we know that the specific abundance of those TIM17, TIM23s regulates the rate of import. And also protein turnover, like I said, is really important. But we also are finding out that phosphorylation of proteins, yeah? It seems to be a really amazing mechanism of regulation. If you phosphorylate something, it's activated or deactivated, or it combines specific peptides or not. So it's becoming more and more obvious that a lot of receptors on the outer membrane of the chloroplast specifically, and the mitochondria, are phosphorylated. Okay, that's it. You survived. That was the end of protein targeting or trafficking in a cell. I know a few of you got Journal Club, so if you have any questions for me, I'll stay back and we can go through your Journal Club questions. Hopefully you all read the paper, so you really have a list of questions, or not. So don't panic about, I'm not going to ask you specifics of these experiments and things like that for your exam, yeah, that's not the point of it. I more want you to understand what methods we can use to characterize a protein import component. I don't want you to talk about specific, figures that I showed, but just understand how you could do that. So you can also use a lot of your knowledge from other lecturers as well, yeah, because it's all very much related. Right. Does anyone have questions regarding the Journal Club? Because you guys have got another lecture here now as well, haven't you? Do you want to just come and see me at a later date? If you have any questions. Who have you got next? Who? Shui. Heng. All right. So email me if you guys have any questions for the Journal Club.